Good day, this is Professor Will, CBMD, PhD. Today is Friday, March 26, 2021. It is 3:02 and 33 seconds, p.m. Eastern Standard Time. Today's research, Nanotech cancer. So let's hop to it here. New Nanotech gives boost to detection of cancer and disease. Date March 25, 2021. Source, University of Central Florida. Summary. Early screening can mean the difference between life and death in the cancer and disease diagnosis. That is why researchers are working to develop a new screening technique that is more than 300 times as effective at detecting a biomarker for diseases like cancer than current methods. The technique uses nanoparticles to increase the sensitivity of ELISA, uses nanoparticles with nickel-rich cores and platinum-rich shells to increase the sensitivity of an enzyme-linked immunosorbent assay ELISA. ELISA is a test that measures samples for biochemicals, such as antibodies and proteins, which can indicate the presence of cancer, HIV, pregnancy, and more. When a biochemical is detected, the test generates a color output that can be used to quantify its concentration. The stronger the color is, the stronger the concentration. The test must be sensitive to prevent false negatives that could delay treatment or interventions. In the study, the researchers found that when the nanoparticles were used in place of the conventional enzyme used in an ELISA, peroxidase, that the test was 300 times more sensitive at detecting carcinoembryonic antigen, a biomarker sometimes used to detect colorectal cancers. And while a biomarker for colorectal cancer was used in the study, the technique could be used to detect biomarkers for other types of cancers and diseases. Colorectal cancer is the third leading cause of cancer related deaths in the U.S., not counting some kinds of skin cancer. And early detection helps improve treatment outcomes. The increase in sensitivity comes from nickel-platinum nanoparticle mimics that greatly increase the reaction efficiency of the test, which increases its color output and thus its detection ability. Peroxidases found in the horseradish root have been widely used to generate color in diagnostic tests for decades. However, they have limited reaction efficiency and thus color output, which has inhibited the development of sensitive diagnostic tests. Nanoparticle mimics of peroxidase have been extensively developed over the past 10 years, but none have achieved the reaction efficiency of the nanoparticles developed. This work sets the record for the catalytic efficiency of peroxidase mimic. It breaks through the limitation of catalytic efficiency of peroxidase mimics, which is a long-standing challenge in the field. Such a breakthrough enables highly sensitive detection of cancer biomarkers, when the ultimate goal is saving lives through earlier detection of cancers. The next step for the research are to continue to refine the technology and apply it to clinical samples of human patients to study its performance. The researchers hope the technology can be eventually used in clinical diagnostic laboratories in the near future. Good day, this is Professor Will, CBMD, PhD, on microbiology, bioinformatics, and pathophysiology. This video is strictly made for educational purposes only. Well, I hope you enjoyed listening. Until next time, have a great day.